Greetings everyone from Vishwakarma University School of Pharmacy. Here I am Professor Sonali Manvetkar. Along with me, Professor Makran Puri sir presenting comprehensive compilation on monoclonal antibodies. And the title for the same is Therapeutic Monoclonal Antibodies, Savior for the Life-Threatening Diseases. Under this topic, I am going to cover introduction of the monoclonal antibodies, type of monoclonal antibodies, discovery, and development of monoclonal antibodies, FDA approved monoclonal antibodies for recent pandemic of COVID-19. So what is the monoclonal antibodies? So antibodies derived from a single clone of plasma cell known as monoclonal antibodies, all having the same antigen specificity, that is produced against a single epitope or antigen. The antibody is also called immunoglobulins. So immunoglobulin is a glycoprotein that is made in response to an antigen and can recognize and bind to the antigen that causes its production. It protects us from microbial infection. They are gamma globulin and synthesized by the plasma cell. They constitute around 25 to 30% of total serum protein. Antibodies are present in serum, tissue fluid, and mucosal surfaces, and on the surface of B cells, where they act as the antigen receptors. Now coming to the structure of monoclonal antibodies, they are composed of 4 polypeptide chains, 2 identical light chains, and 2 identical heavy chains, linked by a disulfide bonds. Light chains similar in all immunoglobulins. Light chains occur in 2 varieties, kappa and lambda. Kappa chains are more frequently found. Heavy chains like gamma, alpha, delta, and epsilon. One Ig contains 1 type of light chain and 1 type of heavy chain. Fc, that is fragment crystallizable, which is fixed component, and FAP, fragment antigen binding, antigen. Now coming toward how it is produced in vitro labs. So the principle behind production of monoclonal antibody in lab, it is mainly with the help of hybridoma technologies. So what exactly the hybridoma technologies? So cloning of B cell stimulate against a single epitope of antigen fused with an immortal cell, that is myeloma cell, to produce a hybridoma cells. So here this mouse is challenged with the antigen, and over the 72 hours after that, the spleen is removed. So spleen is full of B cells. Now this spleen, which is full of B cell, is combined with the myeloma cells. So myeloma cells are nothing but the cancerous cells. So they allow it for the fusion. So after the fusion of B cell and myeloma cells, there is a formation of hybridomas. Okay, so here there is the chances of some of the unfused spleen cell, some of the unfused myeloma cells, and the chances of hybridomas, which is the fusion of B cell and the myeloma cells. So to separate out complete hybridoma cells, it is now cultured with the HAT medium, which is nothing but the hypoxanthine, aminotrypsin, and tyramide. Okay, so once this treatment is happened, now there is a single or the harvested or the hybridomas are separated. So these hybridomas are very peculiar for which is like produce monoclonal antibodies of the same antigen specificity, because due to the B cell and component, and multiplies indefinitely produce the clone of identical cell due to the immortal cell components. Okay, so now this harvested monoclonal further infused into animal, and then further multiplication will take place. So there are the different methods with which this monoclonal antibodies are produced, like phase display, transgenic mouse, B cell immortalization, and single B cell cloning. So there are the different types of therapeutic monoclonal antibodies, like murine, which is 100% mouse, then chimeric, which is a 25% component of the mouse, then humanized, which is the only 10% is from mouse, and then human, which is 100% from human. So now coming based on their different types, their different names are given, like Fab, map, variable fragments, and so on. Then coming toward the discovery and development of the monoclonal antibodies, it is started during the 1970s. And the discovery of this hybridoma technology received the Nobel Prize as well. In 1985, there was the face display technology was launched. In 1986, the very first antibodies was approved by the FDA. In 1994, transgenic mice with the human IgE gene and FDA approved the first Fab fragment. Then during the 2000, FDA approved the antibody drug conjugate and also first humanized antibodies. During 2014, FDA approved the first bispecific antibodies, and 2021, more than 100 monoclonal antibodies approved by the FDA for different clinical conditions. Now, how this monoclonal antibodies mainly shows its effect, that mechanism of action of monoclonal antibody. So they are mainly work with the principle of like suppressing immune system, kill or inhibit malignant cell, also inhibit the angiogenesis. Apart from this, they also show with direct binding effect, complete dependent cytotoxicity, FcR gamma mediated mechanism. So mainly the principle behind this are neutralizing principle. So once any pathogen enter inside the body, that is considered as the antigen. And in response to that, our body generate the antibodies. So we are giving, when there is an immunocompromised a patient is there, so additionally we have to provide the antibodies, and these are nothing but the monoclonal antibodies. So with these are mainly work in the principle of neutralizing capacity. So this monoclonal antibodies are used therapeutically in different conditions, like they are mainly used for the inflammation, like different inflammatory condition, allergic diseases, and the very important cancer condition. So different inflammatory condition like rheumatoid arthritis, Crohn's diseases, then asthma, and then some of the rejection during the surgery, and also the deadliest form, that is cancer. Right? So many monoclonal antibodies are right now in the market. So they are very important function as well, along with their specificity to the disease condition. They do have the very good market values. Okay, so if the pharmaceutical companies are manufacturing monoclonal antibodies, so they do possess the 100% profit in the production of monoclonal antibodies. Right? So some of the examples of monoclonal antibody which are already in the market, like trastuzumab. So trastuzumab is mainly used for the in the cancerous condition, and mainly in the breast cancer. So during breast cancer, there is a too much expression of HER2. So HER2 is nothing but the human epidermal growth factor receptor. So this receptor is overexpressed during the breast cancer. So trastuzumab is mainly neutralizing this HER2 receptor by internalization and degradation of HER2 receptor, disrupt the receptor dimerization, disrupt downstream signaling pathway. It also arrests the G1 phase of multiplication and reduce the proliferation. It also induces the apoptosis with which the cancer cells are dying. It also suppresses the angiogenesis, stimulate the natural killer cells, and inhibit the DNA repair. So trastuzumab is launched in 1998, and it is really showing very promising effect in the cancer treatment by different mechanism of actions. So already I have listed the mechanism with which trastuzumab is showing its effect. Along with apart from this, other monoclonal antibodies like adalimumab, which is mainly used against TNF alpha, infliximab, omalizumab, which is against IgE, daclizumab, puromumab. Okay, so all these are mainly used against the rheumatoid arthritis, Crohn's disease, asthma, and during the rejection of kidney transplant. Also, like some of the anti-cancers like rituximab and all, those are also used for the cancer treatment along with the trastuzumab. And some of the inhibitor of angiogenesis like bevacizumab and abemacicizumab, which is also very which also show very prominent effect in the clinical condition. Apart from this, another example like irenumab, which mainly block the calcitonin gene-related peptide and block the activity of CGRP. Burosumab, which is IgE1 monoclonal antibody, which fibroblast growth factor and reduce the loss of phosphate from the kidney and other metabolic abnormalities. So apart from this, recent pandemic like COVID-19 pandemic. So in this pandemic, we have learned like we need some of the treatment which shows the effect very frequently, very immediate effect. So like COVID-19, it is a very novel pandemic or novel disease where the treatment is not known by the scientists, as there is a lack of clinical trial. So this COVID-19 mainly happening due to the coronavirus, which is very peculiar in their structure, which is having the spike protein on the surface. So with the help of this spike protein, it enter into the human cells. And once it enter into the human cell with the help of translation, it release its genetic material, and that genetic material get replicated, and other factor like spike protein envelope mitochondria, everything get multiplied inside the cytoplasm. And once this translation process over, then further process is assembly of all this spike protein envelope, everything takes place inside the cells, and then release of a virus is happening in the cell membrane. So in this way, this virus is getting multiplied in the host cell, in the human being, and it damaging the further organs. Right? So it is mainly entered inside the body with the help of either nostril or oral cavity, and then it get multiplied inside the body. So when these multiplications are taking place, then with the help of our immune system, our body try to neutralize this virus. But the person, those who are immunosuppressant, those are not producing enough immune cell to neutralize this virus. So in response to that, we have to give additional monoclonal antibody, external monoclonal antibodies. Here, one more thing, when the virus is getting multiplied, there is a production of cytokines. There is a too much cytokines are produced, and due to the too much cytokine storm, they damage the vital organs like brain, lung, heart. And so because of that also, the person is, those who are having comorbidity cannot survive for the long, because there is a damage of vital organ. So here it is a very emergency to develop the treatment where this multiplication of virus will be stopped. There will be the production of the cytokines will be blocked. So here the emergency of developing monoclonal antibody is again observed. So the intense research for the potential novel treatment, including the reuse of the existing drug approval for the this other indication, and the rapid development of the large number of severe acute respiratory syndrome coronavirus to neutralizing monoclonal antibodies are needed. So within a 16 months, 6 monoclonal antibodies have been developed and received an emergency use authorization by the United States or the South Korean regulatory agency, and several additional ones are being evaluated in the phase 3 clinical trial or currently seeking an emergency use authorization. So importantly, these monoclonal antibodies are being tested in different clinical settings and targeted population with different endpoints. So in prophylaxis as well as the early or late disease therapeutic setting. So in addition, monoclonal antibodies in development display a variety of mutation in the constant Fc region aimed to enhance and eliminate effector function or improve the monoclonal antibody half-life and are being utilized as monotherapy or cocktail. So the drugs or the monoclonal antibodies which are given during the pandemic or for the COVID-19 patient is the casirivimab and imdevimab. So a combination of casirivimab and imdevimab, a monoclonal antibody cocktail developed by the Regeneron, is recommended for the patient with confirmed non-severe COVID-19 at highest risk for the hospitalization, and those at higher risk are typically Individuals older than the 60 years and have a chronic disease are incorporated or are non-vaccinated. Also, the patient with severe or the critical COVID-19 who do not have antibodies to the COVID-19 viruses, this drug should be administered by the healthcare worker in a monitored clinical setting along with the current standard of care for COVID-19, where it includes the oxygen and other medication. Casirivimab and imdevimab are a combination of two recombinant human antibodies that targets a different part of the spike protein and the part of the coronavirus responsible for the infecting human cells. So dose of this is for patient with non-specific COVID-19. The total dose of casirivimab and imdevimab is 1200 to 2400 mg given once intravenously. Alternatively, a patient may receive a total one-time dose of 1200 mg subcutaneously. For patients with severe or critical COVID-19, the total dose of casirivimab and imdevimab is 2400 to 8000 mg given once intravenously. So this is the very useful monoclonal antibody which are given during the pandemic situation. So these are the references I have referred during the compilation. So with this, I just would like to thanks. So thank you, Imccrt, for giving me opportunity to present here. Thank you.